Hello, my name is Narangerel, and I'm honored to present my research as part of the Sensors Award acceptance. Before going into my presentation, I'd like to briefly introduce myself. So I'm currently a postdoctoral fellow at Kansai University, focusing on DNA origami, AFM imaging, and AFM nanomanipulation. So before this, I was especially appointed researcher at Osaka University in Japan. And my research mainly focused on K-shaped proteins, aptamers, protein adsorption. And before that, I completed my first postdoc at University of Liège in Belgium. And my research focus was on single molecular nanospectroscopic study and molecular machines. And I obtained my master and PhD degrees at Tokyo Institute of Technology in Japan. And I had my minor degree at also at the same university on focusing on international development engineering and ICT. And I had my bachelor degree at National University of Mongolia. So today I will be discussing my previous work, which was at Osaka University, and this work was nominated for this Sensors Award. So this work titled Measurement of Absorption of Mutant DPS on Carbon Surface using QCM. And this work also funded by CREST, and we recently published a paper on this research in Biophysica in 2024. And here's the outline of my talk here. So I will first give you an introduction of the study, including background and the research goal. Then I will show you the experimental approach or the tools that I used in this study. And after that, I will move on to the experimental results and discussion. Finally, I will finish the presentation by summary. So the study of selective protein adsorption on various solid surfaces is essential for the application of proteins in fundamental engineering fields. Since most proteins are amphiphilic and inherently surface-active molecules, the mechanisms of adsorption are diverse and difficult to elucidate. However, underlying mechanisms of protein adsorption to solid surface is very important issue that is unavoidable for many research fields, such as in biomaterial science to make advanced materials and medical devices and biomedical implants, and in biosensor study for drug delivery, disease detection, and also in food processing, particularly in meat processing and the wine and beer making. So because excessive adsorption can lead to health and sanitation issues, equipment damage. In some cases, protein adsorption used to improve food quality, as is the case in fining of wines. So in nanofabrication technologies, recently researchers are interested in using protein supramolecules to construct complex functional nanostructures for flexible, improved quality of electronics and devices. So we recently proposed a novel material design concept that improves the thermoelectric performance of CNT composites by controlling both thermal and carrier transports at CNT, carbon nanotube, and K-shaped protein, DPS, and carbon nanotube heterojunction using K-shaped protein DPS, and carbon nanohorn binding 12 amino acid peptide aptamers have successfully obtained by biopanning and genetically modified on the DPS outer surface at N and C terminal. And the heterojunction were produced spontaneously and when mutant DPS and CNTs were mixed. So in order to realize more complex and inorganic and biomolecule conjugate with sophisticated functions, and detailed study of the adsorption between carbon surface and aptamer modified molecules is necessary. So this study aims to improve in semiconductor device performance by incorporating cage-shaped proteins into the carbon nanotube network. And aptamers are stable single-stranded DNA, RNA, or peptide molecules capable of binding to its target molecule with high affinity and specificity. And surface modification with them offers very promising perspectives to stimulate the protein adsorption. Protein cages are often spherical, self-assembled, monodispersed, three-dimensional structures. Other shapes such as rods, rings, and more complex geometries are also common. And the protein cage family consists of virus-like particles, proteins, and DPAs, to name a few. And they self-assemble from multiple copies of single or just a few. And the interior cavity is typically used to confine cargo molecules, and it is physically segregated from the external environment. In the case of viruses, nucleic acids are sequestered on the interior of the cage. And the DPS, the DNA binding protein from striped cells, is capable of providing protection to cells during the exposure to severe environmental assaults, including oxidative stress and nutritional deprivation. And cage-shaped proteins can be modified with aptamers through their outside. DPS is selected many studies. In many studies, because of their better structures, which display the both carbon and nitrogen terminals outside of their surface. In this work, we use DPS, which have a potential with inner and the outer diameters of 4.75 nanometer and 9.5 nanometer respectively. The aptamer peptides, 12 amino acids of these sequences that have an affinity to bind carbon materials were genetically added at the two different positions, N and C terminus of the DPS subunit through this linker. And these are here we refer as Y1NT and Y1CT, as you see in the figure. The goal of this work to gain insight into adsorption behavior of cage-shaped proteins with and without newly selected aptamers on carbon surface, and which is crucial for understanding of interaction between cage-shaped proteins and carbon materials. So in order to reach this goal, we aim to establish the method to deposit carbon surface on QCM sensors, and to study the adsorption mass and kinetic of cage-shaped proteins on carbon-coated sensors using QCM, batch mode, and open flow methods. So now I will talk our experimental approach to reach these goals. First, in order to prepare the surface, we use the carbon coating vacuum equipment from KDE. The equipment used a plasma processing function to deposit carbon on the QCM gold sensors, and KDE's vapor deposition source is fibrous, and the current flows easily, and the coating can be performed at a low current and in a very short period of time. So basic steps are: first, we set the carbon fibers between the electrodes, and the fibers are ultra-fine soft fibers, and the vapor deposited carbon particles are coated with a fine particle size. So equipment has preheating function, which applies low heat for 10 seconds to remove impurities from the surface. So after preheating, we rotate the shutter and to separate it from the electrode for the coating. So coating, as I said, can be performed very in short time, like 1.7 seconds. And after deposition, we simply use the medical water rather than using strong washing chemicals to rinse and remove the unnecessary carbon fibers deposited. And after preparing the surface, we studied the absorption behavior of selected proteins on the deposited surface using quartz crystal microbalance, which is QCM in short. So this is from Seiko EG&G. And QCM is well-known and very straightforward method for detecting the absorption of proteins onto a matter surface by measuring differences in the oscillating frequency of the piezoelectric quartz crystal. And the basic concept of the technique is the quartz crystal oscillates constantly by applying AC voltage to gold electrodes. With an AC current between the electrodes of a properly cut crystal, and standing shear wave is generated. So if material absorbs on the gold electrode on the order of nanogram, the wavelength will increase and frequency will decrease according to the mass change in mass. So our QCM uses 80 cut crystal unit with 5 millimeter electrode with a fundamental frequency of 9 megahertz. The amount of absorption can be calculated through this equation called Surbury equation. So we use these parameters to calculate the frequency changes and then convert it to the change of mass. So as I mentioned in the previous slide, QSIM has two measuring methods, batch mode and open flow methods. So here you can see the typical image of the batch mode measurement. So first, a measurement chamber was filled with molecular water to stabilize the system. So here we obtained the baseline here. Then protein was injected here, and as mass is deposited on the surface of the crystal, the thickness increases, and consequently the frequency of oscillation decreases from the initial value. So after a few minutes, when the resonant frequency was stabilized, protein solution is taken out, and the molecular water was injected again for rinsing here. The changes in the resonant frequency and in energy dissipation were monitored continuously during this process. The differences in their values before the injection of the proteins and after the rinse is calculated as absorption masses here. So in the QCM open flow measurement, during the operation, same as batch method, the measurement chamber was filled with the molecular water to stabilize the system, and after stabilization, the proteins were injected and the frequency change has been recorded continuously. For the open flow measurement, no rinsing has been carried out. So the cessation constant, which is a KED of molecules on the surface, can be kinetically evaluated from this open flow experiment. So now let me move on to the experimental results. We observed carbon-coated surfaces by scanning electron microscope first. As you see in this image, the gold surface was uniformly covered by carbon layer. The gold surface morphology could be observed scarcely through the carbon layers. And this slide shows the typical results of QCM batch mode experiments for each protein adsorption. And after stabilization, QCM resonance frequency decreased upon protein injection in all cases, indicating the protein adsorption. And resonance frequency increased by rinsing with murky water, and delta F is obtained by subtracting the average point of frequency after rinsing from the average point of baseline. So 3 cycles of rinsing and injection with protein have been performed, and 3 delta F values have been obtained and averaged and used for calculation of delta n following the Survey equation. So there's a clear differences can be observed between the adsorption of proteins with aptamers, Y1CT and Y1NT, and proteins without aptamers, DPS and CDPS. For aptamer modified proteins Y1CT, Y1NT, frequency change was found to be at these values, respectively, and while DPS and CDP has changed of these values. So in order to ensure reproducibility of the result, we repeated the experiments 10 times for each protein and used average frequency change for data analysis. And the comparison of average mass change is shown in this graph. And Y1NT showed the highest adsorption value, which is 47.4 nanogram per centimeter, and followed by Y1CT and CDPS and DPS. And the native DPS, which had no aptamers displayed, shows very little or almost no frequency changes, but CDPS shows a little bit higher absorption than the DPS, probably due to its carbon nanohorn binding peptide aptamers. But it still shows the less absorption than Y1NT and Y1CT. So in open flow measurement, after the stabilization, proteins were injected and frequency change has been recorded continuously. So in this figure shows the differential curves corresponding to the changes in resonance frequency delta F of the QCM crystal as a function of time. So when we inject this amount of concentration of protein solutions, the resonance frequency decreased in different manners. So basically, frequency change was bigger as protein concentration increased. And in the case of DPS and CDPS, it displayed no frequency change, so which indicated that the DPS and CDPS have lower affinity to carbon surface. And Y1 aptamer on the outer surface of DPS had a strong affinity to carbon surface and though the binding ability to the native DPS. So further, we determined the KD, which is the dissociation constant, from the binding amount and the frequency change using QCM software. So after entering some parameters like molecular weight and sample concentration, we found the dissociation constant values. And KD, which is a dissociation constant, was found to be 0.74 micromolar for Y1NT with very good linearity of determination coefficient and 1.04 micromolar for Y1CT. So it was very notable that the KD was smaller for Y1NT, which shows the higher binding affinity in agreement with the batch mode result that shows the Y1NT had the highest absorption value. So we hypothesized that the position of the aptamer on the K-shaped proteins dictated rates of protein absorption. So Y1 aptamer connected to the N terminal of the DPS works more efficient than the C terminal position. So for CDPS and DPS, it was very difficult to calculate the calculate the KD due to its small change of resonance frequency in open flow measurement. So here we further want to know how much DPS can cover the QCM sensor fully. So as you see, we put here the parameters for QCM sensor and DPS molecules. So using closed packed area equation, we found how much DPS molecules can cover fully the QCM sensor. And so here you can see when we increase the concentration of the protein, and you can see the delta F values, and we found that at 1 milligram per milliliter concentration, QCM sensor is almost fully covered by the protein molecules. So here I will finish the presentation by summary. So in batch mode measurement, the amounts of Y1NT and Y1CT observed on carbon coated sensors were higher than the DPS and CDPS. In open flow QCM measurement, Y1NT, Y1CT observed rapidly, while DPS and CDPS displayed no big frequency change, which is a good agreement with the batch mode measurement. And consistency between these two methods proved that the Y1 aptamer on the outer surface of the DPS had a strong affinity to the carbon surface. And with the regards of dissociation constant, KD, the Y1NT onto carbon surface was more rapid than the Y1CT, which suggests that the Y1 aptamer connected to the N terminal of the DPS works more efficient than the C terminal. So finally, our QCM measurements provided experimental verification on ability of newly selected aptamers, which can increase the binding affinity of DPS to the carbon surface. So our next plan is we are planning to use the new aptamer connected proteins Y2CT, Y3CT. So these will be studied by the same method. So thank you for your attention, and this work has been supervised by Professor Ichiro Yamashita from Osaka University. I'd like to express my gratitude to the Professor Ichiro Yamashita and the lab members Han-san and Chu-san and Yamada-san, and thank you.